Megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell MMC in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division. What is the importance of MMC undergo meiosis? Meiosis results in the production of 4 megaspores, figure 2.8a. Female gametophyte. In a majority of flowering plants, one of the megaspore is functional while the other 3 degenerate. Only the functional megaspore develop into a female gametophyte embryo sac. This method of embryo sac formation from a single megaspore is termed monosporic development. Let us study formation of the embryo sac in a little more detail. Figure 2.8b. The nucleus of the functional megaspore divide mitotically to form 2 nuclei which move to the opposite poles, forming the 2 nucleate embryo sac. 2 more sequential mitotic nuclear division results in the formation of the 4 nucleate and later the 8 nucleate stage of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclear, that is, the nuclear divisions are not followed immediately by cell wall formation. After the 8 nucleate cell stage, cell walls are laid down, leading to the organization of the typical female gametophyte or embryo sac. Observe the distribution of cells inside the embryo sac, figure 2.8b, C. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the central cell. There is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergid have special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tube into the synergid. 3 cells at the chalazal end are called antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac at maturity, the 8 nucleate is 7-celled. Pollination. In the preceding section, you have learned that the male and female gamete in flowering plants are produced in the pollen grain and embryo sac, respectively. As both types of gametes are non-motile, they have to be provided together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grain shed from the anther to the stigma of a pistil is termed pollination. Flowering plants have evolved an amazing array of adaptation to achieve pollination. They make use of external agents to achieve pollination. Can you list the possible external agents? Kinds of pollination. Depending on the source of pollen, pollination can be divided into 3 types. Autogamy. In this type, pollination is achieved with the same flower. Transfer of pollen grain from anther to the stigma of the same flower, figure 2.9a. In a normal flower which opens and exposes the anther and stigma, complete autogamy is rather rare. Autogamy in such flower requires synchrony in pollen release and stigma receptivity, and also the anther and stigma should lie close to each other so that self-pollination can occur. Some plants such as viola, common pansy, oxalis and camelina produce 2 types of flowers: chasmogamous flowers, which are similar to flower of other species and exposed anthers and stigma, and clistogamous flowers which do not open at all. In such flowers, the anther and stigma lie close to each other. When anther dehisces in the flower buds, pollen grain come in contact with the stigma to effect pollination. Thus, clistogamous flowers are invariably autogamous, as there is no chance of cross pollen landing on the stigma. Clistogamous flowers produce azure seed set even in the absence of pollinators. Do you think that clistogamy is advantageous or disadvantageous to the plant? Why? Gytonogamy. Transfer of pollen grain from anther to the stigma of another flower of the same plant. Although gytonogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy, since the pollen grain come from the same plant. Xenogamy. Transfer of pollen grain from anther to the stigma of a different plant, figure 2.9b. This is the only type of pollination which during pollination bring genetically different type of pollen grain to the stigma. Agents of pollination. Plant use 2 abiotic, wind and water, and 1 biotic, animal agents to achieve pollination. Majority of plant use biotic agent for pollination. Only a small proportion of plant use abiotic agents. Pollen grain coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for these uncertainties and associated loss of pollen grains, the flower produce enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollinations. Wind pollination also requires that the pollen grain are light and non-sticky so that they can be transported in wind currents. They often possess well-exposed stamens so that the pollen are easily dispersed into wind currents. Figure 2.10. And large, often feathery stigma to easily trap airborne pollen grains. Wind-pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is the corn cob. The tassels you see are nothing but the stigma and style which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledons. As against this, you would recall that water is a regular mode of transport for the male gametes among the lower plant groups such as algae, bryophyte, pteridophyte. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need for water for the transport of male gametes and fertilization. Some examples of water pollinated plants are Vallisneria and Hydrilla, which grow in freshwater, and several marine seagrasses such as Zoostera. Not all aquatic plants use water for pollination. In majority of aquatic plants such as water hyacinth and water lily, the flower emerge above the level of water and are pollinated by insects or wind, as in most of the land plants. In Vallisneria, the female flower reaches the surface of water by the long stalk, and male flower or pollen grain are released onto the surface of water. They are carried passively by water current. Figure 2.11a. Some of them eventually reach the female flower and the stigma. In another group of water pollinated plants such as sea grasses, female flower remains submerged in water and the pollen grains are released inside the water. Pollen grain in many such species are long, ribbon-like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from wetting by a mucilaginous covering. Both wind and water pollinated flowers are not very colorful and do not produce nectar. What would